I'm very pleased to announce our first speaker to you now, Dr. Keith Ligon from Harvard Medical School. Thank you so much for being online with us today.
Thank you, and to everyone who's joining. I am excited to talk about some of the specific areas that we've focused on over the years with respect to cancer models, cell lines, and particularly 3D cancer cell line models. And our recent work that we've done kind of focusing on how these models can be extremely helpful towards therapeutic investigations in order to find new treatments. Just to briefly go over types of patient cancer-derived models, there's a number of different terminologies and names that people use, so I like to start with just sort of saying that we're going to focus today, for the most part in my lecture, on primary cell cultures and cell lines, organoids or spheroids. There's many different terminologies for this, but the key being that the three-dimensional structure of the organoids or spheroids in brain tumors as well as in other cancers, as was stated earlier, is felt to really help maintain many of the key properties both for science as well as for clinical translation of scientific findings into better treatments for patients. Many of these models are exploding in the field, and hence we have a seminar about it. And so I just like to point out that, growing such models, it really is a growth industry, and is very exciting for all of our labs who have been studying it. Brain tumors are particularly interesting from the standpoint of the heterogeneity and diversity that modeling is trying to represent. Within the brain, there are more than 200 different recognized types of tumors and diagnoses present. And this includes gliomas, such as astrocytomas. The majority of brain tumors that are primary are glioblastoma, up to 75% of them that occur in the brain. But there's also rarer tumors that are frequent, a little more treatable, and have longer-term survival, so end up being disproportionate in the modeling spheres, such as oligodendroglioma, and the needs for studying those tumors which are driven by IDH, as well as rare pediatric tumors like ependymoma. Non-gliomas, there's meningiomas, medulloblastomas, tumors of pituitary organ, which is attached to the brain, and lymphomas. And so this diversity is something that really is a challenge to try to represent. It requires sort of a larger scale than maybe we've had in the past, historically, over the past 50 years. And this doesn't also take into account that for brain, metastatic tumors are actually the most common type of brain tumor. And so within that sphere, lung, breast, and colon are very commonly metastasizing to brain, have potential specific pathways and specific genotypes that would grow within the brain, are particularly refractory to treatment. And this is highly relevant because about 15% or maybe even higher every year of cancer patients overall have metastatic brain tumors. So these are both categories of tumors are ones that we try to study and develop models as best we can for them. And again, this is a major impact. Just briefly, glioblastoma, for instance, is one of the most common cancer killers of the brain, and is incurable. It's also, brain tumors have now surpassed leukemia as the #1 cause of mortality due to cancer in children. CNS diseases are very prevalent, as people may know, and there's no targeted therapies approved by the FDA currently for primary CNS cancers, even though we've tested many of these in clinical trials. In our opinion, a lot of that lack of prior therapies and success is significantly due to the fact that prior models, historical models, did not well enough represent the biology. So many years ago, we embarked to generate at our institution an integrated approach to cancer genomics as well as modeling. And I just show this slide because I'd like to emphasize that in order to do this type of science and have the opportunities that you're going to hear about today to generate models at scale, you really need an infrastructure and coordination between surgery, pathology, oncology, and other disciplines to really execute such a program. And once you have that in place, then it makes things a lot easier as far as generation of both cell lines, xenografts, and the studies that go with them. We've recently launched upon an additional level of external collaboration, which is the Human Cancer Cell Line Initiative, which Dr. Tuveson is also a member of. Our centers were two of the first centers, along with the Broad, to be the first members of this team. And this is really trying to systematize both the production of models in a way that hadn't been done before on the number of patients and number of types of cancers, as well as their functional studies with collaboration with the NCI through CTD squared and other types of functional study programs. We've had great success over the years in generating cancer models at scale. Just as an example, we've had about 5,000 central nervous system tumor patients consented at our hospital over the past 10 years, say, even for such a rare tumor type. And of this, the laboratory, for instance, has received 2,000 plus samples for model generation attempts. And overall, we've generated now, say, in one disease like glioblastoma, around 103 patient-derived GBM cell lines just in our initial phase of the project alone. And as we'll learn a little bit later, these are generated using cell-free, serum-free conditions, and particularly amenable to growing as a non-adherent culture, which is a spheroid culture, established several years ago by many investigators in the neural stem cell field. And so these spheres grow, as you can see in the picture, as free-floating objects, much like free-floating organoids in today's parlance. And what we've tried to do is collect and use this type of cohort in order to create long-term cultures that can be studied experimentally. So, and when I talk about long-term for our group, this is greater than passage 3 in the spheroid three-dimensional type format. They can be expanded indefinitely as the implication. For brain, past passage 3 is usually the cutoff where we have both a pure culture and also the spheroids and organoids at that point will grow indefinitely 20, 40 passages without major changes as far as we can tell in their phenotype and genotype that would make them less useful at that point. And so overall for this program, it's just important to note that only about half of adult GBM patients are you able to get take in an organoid or spheroid type model. And IDH mutant gliomas, for instance, very interesting subtype of mutant gliomas, only about 10% of certain genotypes like that would be sometimes able to be propagated within cultures. Metastatic tumors tend to have a higher rate, about 40% success, but depend greatly upon which type of metastatic tumor you're studying. And I do like to highlight that no low-grade tumor models are able to be derived from brain tumors at this point in our hands. Just quickly to show the level of fidelity that models have on an individual patient level, if you look on the left, the patient tumor is on the top. In the middle is the GBM spheroid, three-dimensional cell line shown there. And on the bottom is a xenograft derived from those cell line spheroid cultures. And these types of tumors maintain both the morphology as well as the immunohistochemical and lineage phenotypes for glial markers such as OLIG2 seen within the patient. And on an individual patient level, we had known that the genotype also was preserved very accurately. Shown on the right is an array CGH profile showing gains and losses that are very characteristic for glioblastoma and that this is highly preserved. But we had a good deal of questions about whether this might be maintained at scale, not just on an individual or small number of patients. And so Dr. Ramin Barokim, who's at the Dana-Farber and Broad as well, and I collaborated with our groups, along with the Broad and Stu Schreiber's group, in order to try and assess more carefully whether 2D versus 3D cultures, how those might perform in growth and genomic validation, as well as in the pharmacogenomics space of treating those cells and seeing what their responses were. So we entered a subset of our cell lines that we had generated a number of years ago. 34 of these patient-derived long-term growth or patient-derived lines that are the newer three-dimensional spheroid-type organoid models, combine those with existing data from cell lines from ATCC for glioblastoma and other sources, and had 44 of those that have been previously profiled in the CCLE effort, and made a larger GBM CCLE effort, which now totaled 78 glioblastoma cell lines of different types, and performed comprehensive molecular characterization of copy number, whole exome sequencing, gene expression, and protein expression in order to integrate that with response to about 380 drugs, small molecule compounds applied to those, and responses were measured by cell titer glow in a standard profiling type effort. And this was work done by our postdocs, Ruben Farah-Luna and Shakti Ramkasoon. And on the next slide is some of the first results just validating that the models, what the 2D versus 3D looked like in this type of setting on a genomic level. And we were pleased to see that even at scale, the patient-derived glioma cell lines, or 3D models, were very similar to the profiles shown here for copy number that we're seeing for patient tumors in the TCGA. And if one looks at the long term in the middle, patient-derived models, which are the ATCC type lines, those, however, showed statistical significant difference between the GBM patient samples as well as the patient-derived cell lines. And so we feel that these models that have been many, many years older and grown as two-dimensional structures have drifted quite a bit more away from the natural state of patient tumors. This was seen when you combine the data and you look across all the genotypes that are kind of known targets in TCGA as driver events. Things like EGFR amplification, shown at the very top on the left there, are present in all, both the 2D and 3-dimensional models, but as example, the amplification events were much more frequent and got very much closer to the state seen in the actual GBM patient tumors. And this was true. The amplicons showed some variation and a little bit less. As people have published, such as Roel Verhoek and Paul Michel, those have been lost in culture sometimes over extended periods of culture time. Tumor suppressor genes and losses that are not drivers that are not extrachromosomal are, however, maintained fairly well, shown in the middle there across different culture types. One thing that was interesting, although the 3D models showed better lineage and genotypic preservation, what we noticed was that when you integrate larger scale number of models over the whole cohort, you get much better pathway representation just from the scaling up of the number of models represented, at least for mutational pathway activation analysis shown on the right. And those were very closely matching to, in most cases, the patient GBM tumors. Overall, I guess I need to apologize. It looks like this image didn't show up in the porting over to the broadcasting system. But overall, in looking at the pharmacogenomics screening results, what we found was that there were many genetic and non-genetic therapeutic vulnerabilities revealed by such a screen. What was interesting is shown here in the top 20 most active drugs. You'll note that there are many compounds that are not targeted therapies, so to speak, on single gene or mutational type events represented. And those non-genetic drugs actually scored higher overall than targeted therapies in GBM. We were very interested to see if the impression that GBM was a difficult cancer to treat and develop drugs for was sort of supported by this overall data. And so this slide actually shows a comparison to all other cancer cell line types that were present in our broad screening efforts to date across all different types of agents and drugs. And what you can see is that GBM is objectively more resistant, shown by red bubbles, to agents overall than the other cancer types. The most sensitive types being hematologic malignancy, shown on the far left, with many different types of green dots. Interestingly, over on the far right of this image, you can see that the patient-derived, more recent three-dimensional models, shown on the left, had more green bubbles. They were weak green bubbles, but they represented that they had more sensitivities than the two-dimensional models, even in this kind of overall survey of cancer drug space. What was interesting was that looking overall, we saw overall resistance correlated best with P53 mutation status. P53 showed that, in both types of model sets, both the 3D and 2D models, a great correlation with resistance to any type of agent. So this was something that we didn't expect and was very interesting. P53 is a difficult thing to drug currently, and so what we were interested in was looking at the wild-type tumors that had P53 in its wild-type state. In those models, what types of drug approaches were most successful, and what we found was that overall, in the entire set of GBM sensitivity, we found that MDM2 inhibitors were actually the most significant hit. In those, it was the most active agent that correlated with genomic sort of status, and that's seen on the left, for both P53 mutations itself, which were anti-correlated with sensitivity, as well as mutations within the P53 pathway, which were either positive or negative. Things like CDKN1A or P21 expression were highly positive correlations for response to MDM2 inhibitors. And this was functionally able to be validated in the cell lines as well, shown on the right, with hairpin sort of reductions in MDM2 and MDM4. We found that the principles also hold out. Going to the next slide, which may be a little slow. Let's see. This was also able to be validated in vivo within a model that was MDM2 amplified. This RG7388 is a Roche agent that's an MDM2 next generation inhibitor, currently in clinical trials for GBM and other cancers. And what we saw was that we were able to extend the life of animals as well as get even reductions and regressions of tumors shown on the right with this highly potent MDM2 inhibitor in a resistant tumor model like glioblastoma. We've also been studying some of the standard of care effects using our cell line models cohort. And so one thing that we had been trying to address was whether radiation should be more personalized, and was there a differential sort of distribution of response to radiation, which all patients get with glioblastoma currently. Cecile Mir, Mayan Hun, and others in my laboratory conducted a screen, therefore, at the Broad of 35 of GBM of these 3D spheroid models, applying radiation in different doses, and then assessing by cell titer glow the effects of these on the cells in order to see what the intrinsic resistance or sensitivity might be. We saw a great range of sensitivity difference in the GBM cell lines, as diagrammed here and measured by cell titer glow area under the curve, whereby there were there was a sort of even gradient distribution of resistance and sensitivity. We didn't see any great nodal sort of effects, and so we were curious what might correlate genomically with this type of distribution in these 3D models. And what was seen again was that P53 was the top correlation as far as explaining that sensitivity and resistance. P53 mutant lines were highly resistant to radiation, thereby explaining at least part of this differences between patient models. This has been challenging to validate in vivo, as right now we don't have any correlation with survival or other status, but we're doing currently different methods to try and follow up on this. And lastly, I just wanted to touch on a point, which is that we often get questions about whether these cell-intrinsic three-dimensional spheroid models could be very informative for immunotherapy, and how might that be. We were fortunate to work with David Reardon and Kathy Wu recently on a neoantigen vaccine trial, whereby we were participating in the exploratory endpoint where we were to isolate GBM cells and T cells from patients who were going on this trial and developing personalized antigen vaccines. And then these types of cell lines were ones that we were able to try to use to see if we could determine whether or not the T cells, which had recognized specific peptides and antigens from the patient, would actually see those same antigens that were derived from sequencing of the GBM tumors in an ex vivo setting as a sort of immune avatar type of situation. In the patients in this study, our conclusion was that tumor antigen-specific T cell responses were able to be seen, but in the brain tumor recognition by those T cells, that was very weak. And in this one example shown here, which got a little scrambled again, it was insufficient to actually, in the ELISpot assay shown on the right, to show any recognition of the brain tumor cells themselves. However, they did see peptide and were activated by dendritic cells engineered for this. So that's an early study, and many people are exploring ways in which to use these types of models in these ex vivo systems for immunotherapy. So just to conclude for my talk, GBM therapeutic screening, so far trials have really focused on RTK activation and kinases. These are well represented in these 3D models. However, our study really suggests that P53 pathway, both in drug space as well as in radiation resistance, may be a very critical pathway to target more aggressively with MDM2 or other approaches. And then the 3D models, especially at scale, are very highly representative of GBM biology and genetics, and can be used, and probably should be used more even, for immunotherapy type studies, as I demonstrated. I'll just end there and say thank you to all our collaborators at the many different institutions on our efforts. Thank you.
Thank you, Dr. Ligon. Our next speaker for this webinar is Dr. Tuveson. Dr. Tuveson?
Yeah, good afternoon, everybody, or good morning, depending on the part of the country you're in. So I'm Dave Tuveson. I'm the Cancer Center Director at Cold Spring Harbor. And the benefit of being the second speaker is I get to take advantage of all the general knowledge that Dr. Ligon just gave you from his terrific work on brain cancer at Harvard. And we can get right into the main points of the cancer type that I study, which is another one of the lethal cancers that deserves a lot of attention by new trainees such as yourselves. So I'm going to have to ask my colleagues at Science to advance my slides if that's okay. So the first slide, is just my name and where I'm working. The next slide is my disclosure slide. These are companies I give advice to. None of that will influence my presentation today, but it's here for the record. And so the third slide that I have, we'll talk about the two themes that I'll discuss today. And I'm taking as an assumption that you guys are seeing these slides change as I say this. Is that correct, Mike and Jackie?
Yes.
Okay.
We are.
Okay, very good. So I think that these cultures can be used for all kinds of things. And you heard from Dr. Ligon about how you can go deep into the molecular details of cancer using them and actually make the first model systems of many cancer types, and then use that information for clinical applications, with therapies for cancer being an obvious one that many people are engaged in. I want to take a slight step back first. I think that these model systems, when they were developed over the last several decades, and I'd say fine-tuned over the last decade by the Cleavers Lab, what they've allowed us to do is really ask fundamental questions about cancer biology and just biology in general, because they are a way that you can grow cells ex vivo. You can do a variety of reconstitution experiments with additional cell types. You can really, get your hands around parts of mammalian cell biology that, I know of no alternative system that allows it. So if you go to the next slide, it's a little bit of a cartoon of, summarizes what we've done. This is slide number 4, where my laboratory over the last several decades made mouse models of pancreas cancer and we learned all kinds of things from it. But working with the fellow on the right of the slide, who's Hans Cleavers, who's an avid fisherman, we learned how to grow the tumors from the mice and the tumors from the patients using these three-dimensional methods. We call them organoids based upon the nomenclature that Hans's group gave to it. They're dependent upon the presence of a mitogen cocktail, as well as Wnt pathway activation and TGF-beta pathway inhibition in a semi-solid media. The most common media that has been used is an off-the-shelf product called Matrigel that you've all heard of. There are lots of alternatives. There are inexpensive ones like collagen, rat tail collagen type 1, and then there's lots of investigational products that are being used too. The benefit of Matrigel is that you add cold liquids to your cultures and it melts the Matrigel. And so you can actually keep biology stopped or paused while you isolate cells from your cultures and then do your assay of molecular pathways, et cetera. Whereas if you use some of these other matrices, you have to warm it up in order to break the matrix down, which obviously could change many of the parameters you may seek to study. So if we go to the next slide, which is slide number 5, one of the first things we noticed when we made pancreas cancer organoid cultures is that when we cultured them in the presence of a cell type, which is a resident mesenchymal cell in the pancreas called stellate cells, that the cultures developed into a two-cell state system where we had some of the features of pancreas cancer. And so stellate cells are a cell type in the pancreas and the liver. They store vitamin A, and their usual job in the body is to store vitamin A and release it so that you can make retinoic acid and your visual pigments work. When the pancreas or the liver are damaged, they differentiate into a fibroblast type cell population. They proliferate and they start to secrete basement membrane components and matricellular proteins. This is a common feature, desmoplastic tumors of for example, the pancreas and the liver, where it's very highly fibrotic. And so we took stellate cells from a mouse, added them to pancreas cancer organoids, and lo and behold, we mixed them together. As you can see, a matrix was made, which is to the right of the slide, where you see the pink material around the cancer cells. And you can you study the matrix by doing mass spectrometry, electron microscopy, and of course, less sophisticated analyses using antibodies, et cetera. And so this, to our knowledge, was the first real co-culture system that made matrix, and we were really taken by that. And if you go to the next slide, you'll see that in the matrix, we actually realized that the initial stellate cell population had turned into two types of fibroblasts, one of which we call myofibroblasts, which is what others would consider to be a cancer-associated fibroblast. They express smooth muscle actin and the partner myosin protein for that. And these cells are found in close proximity to the cancer cell. So in the upper left of this slide, in the in situ, you see green cells. These are the cells expressing keratin 18. These are the cancer cells in the organoid. Surrounding them is a layer of red cells. By in situ, the probe is picking up alpha smooth muscle actin. Now, above those cells, you see a bunch of blue DAPI-stained cells. Those are actually the stellate cells we started with, but they don't express smooth muscle actin. So what do they express? On the two little micrographs below it, you see that there's now some cells that express IL-6. And this is actually a stellate cell that's starting to make cytokines and chemokines, and IL-6 is one of them. We can find the same biology in tumors from mice and humans. In the upper right, there's some evidence showing that. And on the lower right, it shows that if we were to isolate so-called inflammatory fibroblasts, or ICAFs, and just plate them on a petri dish, rapidly within hours, they become myofibroblasts. They express all the same genes of a myofibroblast, et cetera, suggesting there's a dynamic regulation of these two populations. We never would have found any of this if we hadn't made the organoid cultures in the first place and done these add-back experiments. And I think it'll be really cool for when Keith starts to do experiments like this in the brain, because no cancer happens in isolation. There's a cell that acquires mutations, but the initiation and progression of that cell is highly dependent upon the tumor microenvironment. And again, these types of experiments are very challenging to address. On the next slide, one of the star postdocs in my lab, Julia Biffy, took this analysis to the next level. Just a little cartoon showing you the different types of fibroblasts that we can derive from stellate cells. One we call quiescent fibroblasts, where we just put them in a three-dimensional matrix. They're metabolically active, not particularly proliferative. They actually have gene signatures like a stellate cell you'd have in a resting pancreas. In the middle, we can make inflammatory fibroblasts by exposing them to conditioned media from organoids, so in a transwell type of assay or in a conditioned media transfer, for example. And then to the right, again, myofibroblasts, very simple to generate them, essentially just by allowing them to contact a petri dish where the tension between the cell types appears to be something that helps readjust their differentiation pattern towards a classic cancer-associated fibroblast. So Julia, using this system, actually was able, in the next slide, was able to show the mechanistic underpinnings of these two types of fibroblasts. And so this next slide should show you a cartoon. This is slide number 8, where you have the inflammatory fibroblasts on the left and the myofibroblasts on the right. Inflammatory fibroblasts are yellow, the myofibroblasts are blue. She published a paper about half a year ago showing the pathways that are required to actually generate both cell types simultaneously. Because again, the cool thing about biology is you essentially have different patterns of cell behavior dependent upon the topology, upon where the cells are located in tissue. And it turns out the purple cancer cells in this case are liberating ligands. And the two ligands which are pertinent and necessary for this behavior are TGF-beta and interleukin-1. And both are made by the purple cancer cells, but in this case the TGF-beta works locally because it binds to that matrix, that pink material that was on a couple of slides before, where it's activated into an active form by proteases. So it really doesn't diffuse far away, whereas interleukin-1 diffuses very far. And it turns out in a mutually antagonistic way, the TGF-beta pathway and the interleukin-1 pathway regulate the patterning of fibroblasts, at least in the pancreas cancer models that we're looking at in vitro and in vivo. And the real mechanistic sort of nuts and bolts Western is on the bottom, where interleukin-1, which is present in the conditioned media made by cancer cells, upregulates the expression of its receptor, the interleukin-1 receptor. This is totally antagonized by the presence of active TGF-beta in the culture. And so she published a very nice paper that was, cause and effect, genetics, and cell biology and biochemistry, all allowed by the fact that we had this in vitro surrogate system based on organoids co-culturing it with mesenchymal cells. Now, at the end of Keith's talk, he talked about how he's now excited about doing immunotherapy type experiments, working with Kathy Wu at the Farber, et cetera. And likewise, those type of questions can be asked using these systems too. I've just showed you, just about the fibroblasts so far, but if you were to go to my summary slide on the sort of big biology part of my talk, it's entitled CAPS and PDAC. It should be slide number 9. We basically stumbled into the CAPS subtypes by making these organoid co-cultures. This was not our intention, but this is what we found. And we essentially identified 2 distinct types of fibroblasts, myofibroblasts and what we call inflammatory fibroblasts. And shown briefly to you, these can interconvert from one cell type to the other, suggesting they are multi-potential cells, each having the ability to become the fate of the other. It's not a hierarchical type of differentiation. It's actually a much more dynamic form of cell biology. And this is relevant because these cell types play a role, not just in cell biology and pancreas cancer initiation and progression, but also likely in the response to therapies and the influence of the immune system on responding to therapies as you try to treat cancer. Okay, so I'm going to segue from the fundamental biology that you can develop new insights by using organoids into something that's much more applied and I think related, at least in theme, to some of the things you heard about from Keith. And so the next slide is slide number 10 in my deck. It's survival plots of pancreas cancer patients when they're treated with medicines in the clinic. And these are patients with advanced pancreas cancer. They have the disease has spread from the primary tissue. Oftentimes it's metastatic, about half the time when you meet a patient for the first time. And on the left is one clinical trial, and on the right is another. And each of them show a separation of the curves at the median point, meaning when half the patients are still alive or haven't progressed, but the tails of the curves are very similar, unfortunately, meaning if you were to go out a couple of years, really very few patients are still alive, despite what therapy you use. And so again, on the left, you're comparing a single agent chemotherapy called gemcitabine to a group of 3 chemotherapies, FOLFIRINOX, and on the very right, it's gemcitabine compared to the gemcitabine with paclitaxel that's formulated with albumin. It has a trade name Abraxane. And so these were papers published in the New England Journal of Medicine, which many of you know is where, kind of the highest evidence clinical science papers are published when you're trying to change the standard of care treating patients, and they have impacted how patients are treated in the clinic. And so despite the survival looking as terrible as it does in pancreas cancer, about half the patients who get these medicines in the clinic actually feel better after they get the medicine. The pain and the pressure from the cancer goes down, they eat more, they spend more time with their family and less time seeing their doctors. But none of them are curative. But the question is, if we could actually pick the right chemo to give the patient, be it the FOLFIRINOX chemotherapy or be it the gemcitabine with Abraxane, would that actually benefit more patients and keep more of them healthy for a short period of time while you figure out the next smarter medicine, like you heard Keith talking about in brain cancer. So on the next slide is a summary slide of the study that we conducted in human pancreas cancer using organoids. And so you should see a rather attractive picture of a human standing up with a scalpel pointing into the tumor that's in their pancreas, or a needle pointing into that tumor. So as you all know, the pancreas is a vital organ. You would require it for vitality or life. Its job is to allow you to digest food and balance blood sugar. If you were to look at a pancreas, it'd be like looking at the brain in an adult. You really wouldn't find dividing cells. It's a quiescent tissue. You only find dividing cells if there's damage to the pancreas, and I think that's pretty similar to the brain. And so there's damage, at least in the pancreas, when there's inflammation, and inflammation is caused by external and internal forces. There's a disease called pancreatitis, for example, which we all get all the time, sometimes because we celebrate too much, sometimes because we have gallstones that go the wrong way up our pancreatic duct. But these are likely initiating events in this disease, activating cell proliferation. What we did, though, is we took pancreas tumors from patients. They either had localized pancreas cancer, where they would go for surgery, and about 10 to 15% of all patients get an operation for pancreatic cancer, or we took it from patients who had advanced disease, who had metastatic pancreas cancer or locally advanced pancreatic cancer. We made the organoids, which is a little a picture you see next from the blue arrow, and then we analyzed them. Similar assessment to what you heard from Keith, and here's his cultures. So we, on the bottom, did nucleic acid analyses, looking for mutations in the DNA expression of the mRNA. In the middle, we tested drugs, including the 5 drugs I talked about that we treat pancreas cancer patients with. We also looked at investigational agents, like the ones Keith were talking about. And so on the next slide, this is actually a picture of Hervé Thiriac, who is the lead scientist who did this study, his thumb on the top and a 1.5 cc Eppendorf tube of a small biopsy that you can get from a pancreas tumor from the physician who's trying to diagnose the patient. So you see at the bottom of the tube these little stringy pieces of tissue, and once you lyse the red cells, which is the lower micrograph, you can see the tissue a little more clearly. You embed it in the matrix that I talked about with the factor support, and within a day to 2 days, you will see those structures, which are the organoids that come right out. These are single-cell lumens. Oftentimes, sometimes they can be filled, but very quickly, within a day to 2, you know if the culture is going to work or not, and then you can passage it and ask questions. So on the next slide, this is kind of the report card of how we did this study. We had 78 patients that went for surgery, 60 patients who had advanced pancreas cancer, 1 who recurred, 20 patients who actually participated in a clinical trial where after they died from pancreatic cancer, an autopsy was performed rapidly to harvest tissue from their bodies so that the scientists could learn about this disease. And to the right, we had the pancreatic donors, which in America are usually motorcycle accident victims who are usually a bit younger than all the patients that have pancreas cancer. Their pancreatic tissue is harvested for islets for brittle diabetics as a transplant for diabetes, and we get the rest of the tissue, which includes the acinar cells and the ductal cells from which the organoids are derived. And underneath the number of n equals 78 is how often we were successful. So we were successful 61 times out of the resected patients, and you can walk down the numbers to see our success rate, which is a relevant point to discuss. On the next slide is our approach to the genetics of pancreatic cancer, and it has the genes listed and the frequency of finding it, as well as the stage of the disease. So if you look to the very right on the top, the normals, which were the unfortunate individuals who died in a motorcycle accident, they basically don't have mutations. And if you were to look at the patients with pancreas cancer, they have all kinds of mutations. The one that's very common is the KRAS mutation. This is an unanswered opportunity for those of you on the phone to figure out a way to target KRAS. This is a GTPase. When mutated, becomes a slow enzyme. We have no medicines that speed up enzymes. And so this has been, chemically, theoretically, a problem for the field. The mutation we have in pancreatic cancer is usually a glycine to aspartate mutation or Tuveson. And so the cysteine mutants that you find in lung cancer, we basically don't have them in pancreas. So this is a hard problem, and we got some bright people working on it, maybe one of you. Underneath it, we have P53 mutations. They occur in most of the time, 88% of the time. And I should have mentioned, in the KRAS mutant cases above, 63 out of 66 carry the mutation, 3 don't. And the 3 that don't carry druggable dependencies, and I'll show you a brief example of that at the end. And then there's a variety of other mutations, and CDKN2A, which includes the P16 tumor suppressor gene regulating the cell cycle, SMAD4, which is a participant in TGF-beta signaling. And these are the mutations you see in the exome, okay, so in the DNA. And so we found these levels of mutations, and I'm not showing it here because we don't have time to go through everything, but if you did copy number analysis, you would realize that you had lots of copy number loss of CDKN2A and SMAD4. Much more common to find that than it is point mutations. Okay, going to the next slide, besides the DNA, we did the RNA, and we looked at the RNA in different ways. This is one simple way, just by a principal component analysis. And in black, you see a cluster of the normal pancreatic ductal organoids, and in the various colors, you see them, they're far away from the black ones, and they're not necessarily all in the same bin, but they are distinct. And there's one little cluster to the left, which is called HM19A through D. That's the cluster that came from 4 different tumor harvest sites from a patient who succumbed from pancreas cancer who underwent a rapid autopsy. So you can look at the RNA that way, or you can look at the RNA this way on the next slide, which is slide number 16, showing you 2 different patterns, roughly speaking, of gene expression in the organoids, which at Cold Spring Harbor we called C1 and C2, and they're very similar to 2 different types of pancreatic cancer that were discovered by Andrew Biancan and Peter Bailey, as well as Eric Collison and Richard Moffat and Jen Janier. And, so we can see the 2 types in the organoids. And that's important because one of the types, which in this case is C2, is very uncommon if you were to grow pancreas cancer as a cell model on plastic. C1 predominates, which is the more basal quasi-mesenchymal. And that's true, actually, I'd say, of a lot of cancer types. Lots of cancers, you can grow them on plastic, and they represent a more mesenchymal form of that cancer, but may not be representative of what you find in vivo. And so we had a good collection of the 2 types of the more classical and the more basal, and then we wanted to ask questions. And so on the next slide, it's just a cartoon showing you how we do pharmacotyping, which is another word for drug testing. And we had the 5 types of drugs, the 5 chemotherapies that I mentioned before, and just showing you that you can do drug testing in organoids and matrigel. We use a flat bottom well approach for doing that, where you have a matrigel instead of a dome, just a layer, but it works great. In this case, this is the classic ATP assay for viability. We also have a microscopy surrogate shown here, and I think high-throughput microscopy is certainly the direction that many of us are going in order to do these experiments much faster with less input. On the next slide, though, I would say the meat of the whole thing, which is when you look at chemotherapy, the ones we give patients, there's a tremendous amount of diversity in the response to the cytotoxic drugs the doctors give in the clinic. And we don't think of cytotoxic drugs that way. We think of them as we give as much as we can before the patient gets sick, and we look for the side effects. We really don't dose chemotherapy to any efficacy endpoint, at least in pancreatic cancer. And this is just showing you in chemotherapy-naive organoids, there's a tremendous amount of diversity in the specimens, innate diversity. And, this is pharmacogenetics, if you want to think of it that way, or pharmacotranscriptomics, as you'll see in a second. And so you can separate the organoids into very sensitive, which would be to the left, and more resistant to the right. And we did that, and we came up with a kind of simplistic way of looking at it into tertiles, where blue is sensitive and red is resistant. And then we use that information to ask questions. On the next slide, the way we asked the questions is we asked, was there a correlation between drug response and genetics? And the answer was no. Well, what about drug response and transcriptomics, gene expression? The answer was yes. And so the way we did it was Pascal Below, working with Alex Krasnitz, were very clever, and they were able to actually do this analysis using a variety of approaches, and they could come up with a gene expression pattern that correlated with great response for each of the 5 chemotherapies, because we had so many patients, in our input. On the next slide, we could use that information then to classify our organoids. And so on the right, you see more red. On the left, you could see more blue. And this is just the case for gemcitabine. So we could use the signatures we made to go back into our organoids. And that's good, but it's totally circular. Whenever you make a new assay, you have to go outside of your experimental setting and find another data set. And so we did a cross-validation by taking this information and looking right into human specimens. And on the next slide is a picture of a pancreas cancer H&E slide. You see the cancer cells outlined in red, and you see the stroma, which is outside the red. This is very similar to that organoid co-culture I showed you before. And we were fortunate enough to find a group of people who actually took those cells outlined in red, laser captured them, got high-quality RNA, did RNA sequencing and DNA sequencing away from the stroma, and treated those patients with the drugs that we talked about already in pancreas cancer. So on the next slide, it describes it. This is a terrific group of collaborators from Toronto, Canada. Stephen Gallinger is the lead there in surgery, Jennifer Knox in medical oncology, working with Sandra Fisher, who's a fabulous pathologist who developed many of the methods and actually did the hard work of laser capturing all these samples. And again, I would tell you that if you look at TCGA, you don't have this information. You have like bulk tumor information. And unfortunately, the other challenge with TCGA is we don't have terrific clinical annotation of the specimens in terms of what was the patient treated with, how did they do, et cetera. And so at least in pancreas cancer, we do, fortunately, thanks to our northern neighbors and collaborators. And so we looked into their data set in 2 different trials. The first was where they gave gemcitabine after surgery. So if you go to the next slide, this is again slide number 23, you see how the gem signature separated you on the left to more red, on the right to more blue. But what does it mean? Go to the next slide and you see what it means. So this actually shows that the gemcitabine signature in the patients that got gemcitabine, the left-hand curve, if you got gemcitabine in this case and you had the signature, you're in green, and if you didn't have the signature, you're in blue. And this is progression-free survival, but it's like a 2-fold difference in the median overall survival with an actual tail difference. And again, this is just, these patients got gemcitabine, they had pancreas cancer and went for surgery. So this was kind of interesting, very provocative, strong hazard ratio. Then look to the right. We had 30 patients who we knew the signature information on, but they never got treated. About 30% of patients who go for surgery don't get adjuvant treatment because the disease progresses fast or various other things happen. And this shows you that you can't tell the green and the blue apart. And this was relevant because there's 2 types of biomarkers, predictive biomarkers and prognostic. And we clearly have a predictive biomarker for gemcitabine because it mattered based upon the treatment. So this was very exciting to us, and then it allowed us to do the second question, and then I'll be done in about 2 minutes. The second question was, we had patients with advanced pancreas cancer. I'm on slide number 25, and they received FOLFIRINOX, is the only one I'm going to talk about. and on the same deal otherwise, laser capture microdissection. And on this final slide, it's a waterfall plot. And in a waterfall plot, if you're going down, the tumor's shrinking. You're going up, the tumor's growing. This is 8 weeks after starting therapy. And you see a lot of blue to the left and a lot of red to the right. Well, in this case, if you have the signature for oxaliplatinum, your chance of shrinking is much higher. And if you're lacking the signature, your chance of growing is much worse. And so this really suggested to us that there was something to these chemotherapy signatures, and we're moving that towards the clinic right now. And on the next slide, it shows the clinical trial diagram that we have. We're starting a trial with Jennifer Knox will run it from Canada, and Liz Jaffe and Dan Laharo here in the States. And it's basically to ask, in a randomized setting, when you compare the two chemos, does this predictive biomarker really work? And so we're going to use it in an exploratory fashion this time, and if it looks good, then obviously we'll try to roll it out prospectively. And there's no reason to really go into the details of that. And the last slide that's of relevance is really this picture that you started with, Hervé, and to the right, showing you the two different things we found from doing this study. On the upper right, yes, the DNA signature works for some targeted agents like afatinib. We had 3 RAS wild type. One had an EGFR mutation, ERB2 mutation. It was hypersensitive to afatinib, which is an irreversible ERB inhibitor. So that's interesting, genotype, phenotype. But on the bottom is really the money, because this is a common signature, meaning many patients may have these transcriptomic signatures correlated with drugs. So pharmacotranscriptomics may be a path forward for this disease to give patients that are upfront chemo correctly, so then we could have the more clever therapies for them. And so we've learned a lot about basic science and applied biology. I think I took up more oxygen than I was supposed to on this call, but we still have a few minutes left for your great questions. I want to thank Keith for going first and allowing me to take up more time.